next, sixth question, differential diagnosis of congenital malformation of the lungs. And the last question, we will give more detailed characteristics of the genetics of bronchial asthma, like typical examples of complex or multifactorial disease of the respiratory system. So, disease of the gastrointestinal tract occupy one of the leading places in the general human morbidity. Hereditary disease of the digestive system leads to disruption of various levels of digestion, cavity, parietal and membrane digestion, absorption, and the development of malabsorption syndrome. The cases of the disease which happens in case in gastrointestinal tract depends from the different types of the digestion. There are three main types: cavitary digestion of complex substance or polymers in cavity of small intestine. The next type, membrane digestion of simplex substances on cells membrane of enterocytes, and intracellular digestion, final digestion of substances in lysosomes. And in genetics for hereditary pathology of gastrointestinal tract, we deals with different forms of the pathology which may happen in with the problems in cavitary or membrane or intracellular types of the digestion. Main features of membrane digestion includes very high velocity of food digestion. This process cannot be reproduced in vitro. Complex mechanisms of nutrient digestion at the final digestive stage, and are highly effective cooperation between digestive and transport processes. So, there are a lot of enzymes of intestinal mucus involves into digestion. Many of them: carbohydrates, proteases, esterases, nucleases, and each enzyme is controlled by the special genes. So, in generally, malabsorption syndrome is an alteration in the ability of the intestine to absorb nutrients adequately into their bloodstream. The disorder of intestinal absorption or malabsorption syndrome is the actual problem of clinical gastroenterology and pediatrics, because many gastrointestinal disorder development as a result of malabsorption syndrome. Almost 70 pathological conditions can be accompanied by malabsorption syndrome. With according of the Wilkinson's classification, malabsorption syndrome we made to divide it into there are several types. First of all, the classification of malabsorption syndrome depending on the etiology and type of disorder. The first one include congenital and hereditary forms of malabsorption syndrome. It includes the primary insufficiency of digestive function of the small intestine, for example, maldigestion syndrome, intestinal enzymopathy such as insufficiency of intestinal enterokinases, peptidases, etc. And second one, the primary insufficiency of absorptive function of the small intestine, monosaccharides and etc. So the next secondary form of malabsorption syndrome, of malabsorption and maldigestion together. First of all, enterogenic form, inflammatory bowel disease, for example, enteritis, Crohn diseases, intestinal infection, parasitic invasion, and etc. Gastrogenic form includes in secondary forms of malabsorption syndrome, for example, gastric ulcer disease, chronic gastritis, cancer of stomach. The third form, pancreatogenic form, include chronic pancreatitis, cystic fibrosis, and pancreatic cancer. Fourth form, hepatogenic form, include acute and chronic disease of the liver, hepatic cirrhosis, cholestasis, and etc. Fifth one, after surgical inventation in the gastrointestinal organs, in case with intestinal surgery, may develop the secondary forms of malabsorption syndrome. The next secondary forms, six ones, are endocrine forms in case with diabetes mellitus, hyperthyroidism, and etc. And the last seven forms, malabsorption syndrome may development after medications, after their using of the next several drugs which present on the slides, and then other also may forms. And malabsorption syndrome secondary forms may development after the radiation affection of gastrointestinal tract. Second type, the classification of malabsorption syndrome depending on the clinical stage. First one, latent form. It is diagnosed only by functional tests. Obvious form, it includes there are three stages: initial stage with expressed clinical manifestation, and terminal stage. So, common clinical picture of malabsorption syndrome. Some characteristics includes diarrhea, abdominal distension with cramps, bloating and gas due to impaired water and carbohydrate absorption, irritation from the unabsorbed fatty acids, steatorrhea, edema, malnutrition and weight loss due to decreased fat, carbohydrate, and protein absorption. Weight may be 80 to 90% of usual weight despite increased oral intake of nutrients. Also, common clinical picture includes muscles cramping due to decreased vitamin D, calcium, and potassium levels, muscles wasting and atrophy due to decreased protein absorption and metabolism, perianal skin burning, itching, and etc. Children with malabsorption syndrome often exhibit a failure to grow and thrive. So, the some monogenic forms of their hereditary pathology characterized by the malabsorption syndrome. And first of all, let's give the characteristics of the celiac disease or gluten-sensitive enteropathy. Celiac disease is a malabsorptive disorder characterized by permanent gluten sensitivity enteropathy resulting in malabsorption, failure to thrive, and gastrointestinal manifestation. More genetic characteristics of the celiac disease you may read in OMIM on database online, Mendelian inheritance in men. Don't forget about this database when you will be study information and some materials for the classes, for the practical classes in genetics. Celiac disease, also known as celiac sprue and gluten sensitivity enteropathy, is a multifactorial disorder of the small intestine that is influenced by both environmental and genetic factors. It is characterized by malabsorption resulting from inflammatory injury to the mucosa of the small intestine after the ingestion of wheat gluten or related rye and barley proteins. Prevalence: one individual for 120 or 30 individuals. It depends from their special population and regions in which spreads this pathology. Onset up to 2 years, very early onset. Girls are affected 2 times higher than boys. Gluten sensitivity enteropathy have the autosomal recessive with incomplete penetrance inheritance. So, and number science is used with this entry because accessibility to celiac disease, first of all, celiac first type, has been found to genetically determined by processing of specific HLA-DQ alleles, which localize on chromosome 6, and this region has been designated celiac 1. Also, in case with celiac disease, we need to identify association of the HLA antigens, DQW2, B8, DRS3, and etc. Main clinical picture: gastrointestinal manifestation include failure to thrive, diarrhea, chronic or recurrent, occasionally fulminant, and etc. And of course, malabsorption signs and symptoms. Second manifestation, musculoskeletal manifestation, wasting limbs, but with a marked abdominal destination. And other picture include anasarca, anemia, apathetic, delayed puberty, hypertonic, muscle hypertonia, may development mental retardation, irritable, mouth sore, pale, and smooth tongue, dental enamel hyperplasia, and etc. On the next photos, you may to see the pigment spots at the back and arms of the patient with the celiac disease. Also, eruption in angles of mouth and gingiva. You may to see it here, yeah. Diagnosis include hyperchromic anemia, steatorrhea, dysbacteriosis in small intestine, low blood levels of calcium, magnesium, ferrum, albuminum, total lipids, cholesterol, low density lipoproteins, and but high blood levels of IgE and IgA antibodies to gliadin. And by biopsy, specific morphological abnormalities of small intestinal tissues we need to detect. So, another form of hereditary disease with malabsorption syndrome, it is insufficiency of intestinal disaccharides. Inheritable defects in enzymes, such as small intestinal discardiosis, that show persistent gastrointestinal disorder due to the abdominal hydrolysis and transport of discarides in the intestinal mucosa. On the slide, you need to see the schemes of absorption of nutrients, small intestine, and main stages. Digestion, absorption, monosaccharides, which are present in small intestine. So classification of the inefficiency of intestinal discardiosis includes lactase deficiency, saccharase deficiency, maltase deficiency, isomaltase deficiency, amylase deficiency, and combined deficiency of discardiosis. Also, you need to see that characteristics of this pathology present in OMIM. Disruption of absorption systems and transport of amino acids. Disorders of the absorption system of neutral amino acids includes most amino acids. Also, disorders of the absorption system of major amino acids such as arginine, lysine, and cysteine. The third group, disorders of the absorption system of acidic amino acids, aspartic and glutamic acids. Fourth group includes disorders of the absorption system of proline, hydroxyproline, sarcosine, betaine, and glycine. And fifth group, disorders of the absorption system of methionine. For illustration of the congenital abnormalities of amino acid transport, we need to describe the Laurel syndrome or low ocular cerebrorenal syndrome. Is a rare X-linked multisystemic disorder characterized by the triad of the congenital cataract, several impaired intellectual development, and renal tubal dysfunction with slow progressive renal failure. So this syndrome, Lord syndrome, depends from the mutation in ocular cerebrorenal gene, which localize on the X chromosomes in position in locus 26. So, in other examples, Hartnup disease, characterized by the transient manifestation of pellagra, cerebral ataxia, and psychosis. It caused by impaired transport of neutral amino acids across epithelial cells in renal, proximal tubules, and intestinal mucosa. Hartnup disease is caused by homozygous or compound heterozygous mutation in the special genes ACC6A19. Genes are the genes of Hartnup disease, which localize on chromosome 5. The patient characterized by periodic and reversible episodes of skin rash like presence here on this slide, resemble pellagra, erythema, redness of the skin, scales may present, are located in open areas of the body and are broken by sunlight as well hot weather. The next examples of the congenital abnormalities of amino acid transport, congenital tryptophanuria. In this case, may development hyperplasia of corpus callosum, demyelination of the brain, like present on this photo. So, general conclusion. about the hereditary disease or hereditary condition of amino acid transport. First one, the primary causes of impaired digestive function of small intestine are genetically determined abnormalities in molecular mechanism of nutrient transport. Second, the molecular transport abnormalities arise in membranes of enterocytes and should be considered as a pathology of cell membrane. The presence of selective disorders in small intestinal absorption is an important confirmation for existence of different systems transporting glucose, galactose, fructose, amino acids, lipids, bile acids, vitamins, phosphates, electrolytes, and etc. So, now let's give the more detailed characteristics of the next form of hereditary diseases with malabsorption syndrome. We told with you about the cystic fibrosis or mucoviscidosis. Yeah? Cystic fibrosis is classically described as a threat of chronic obstructive pulmonary disease, exocrine pancreatic insufficiency, and elevation of sodium and chloride concentration in sweat. Cystic fibrosis transmembrane conjugation regulator, it is a gene which involves into the pathogenesis of cystic fibrosis. SFTR gene SFTR is located in apical part of cell membranes and secretory cells and the excretory glands. And all problems in a patient with cystic fibrosis depends from mutation in these genes. Pathogenesis. So the gene mutation in cystic fibrosis involves into the problems of the structure of the cells and decrease the absorption of electrolytes, protein from cavities of glands, and includes into the blockage of ducts by viscous secrete and inflammation development. There are several clinical forms of cystic fibrosis. It's classified by, there are five main forms. The first of all, combined form, affection both digestive and respiratory tract. Approximately 75% of all patients have the combined form of cystic fibrosis. Respiratory form, which in which are present the only symptoms in our lungs, approximately 15%. Intestinal type of form, 5% of all patients. Meconium alleles in 5%. And atypic and latent form, approximately 1 to 4% may have this atypic or latent form. Clinical manifestation involves there symptoms in disruptions, weight loss, obstruction, and etc. Respiratory system includes persistent cough with productive thick mucus, wheezing, shortness of breath, frequent chest infection, sinusitis, nasal polyps, and etc. may development. Also in patients with cystic fibrosis present the reproductive problems. 95% of patient men and 20% of women are infertile. And also development in patient with their cystic fibrosis, development the osteoporosis and arthritis. The next group of disease of gastrointestinal tract such as peptic ulcer and duodenal ulcer, gastritis and gastroduodenitis, like typical multifactorial or complex diseases, pancreatitis, enteritis and enterocolitis, Crohn's disease, non-specific ulcerative colitis, and there are a lot of forms of congenital malformation of the digestive system we need to detect in newborns. Congenital malformation of gastrointestinal tract, like typical common forms of the pathology, depends from the mutation and hereditary materials enduring there and mistakes during the embryonic development, and it depends from the influence their congenital malformation of gastrointestinal tract by etiology. It includes hereditary, exogenous, and multifactorial congenital malformation. For example, in case with the chromosomal abnormalities, 5% detect presence there problems of gastrointestinal tract. Mutations of individual genes or monogenic pathology in 3% cases. Environmental factors cause approximately 2% of all congenital malformation of gastrointestinal tract. And polygenic multifactorial, 90%. Most of all. But in more than 70% of cases of congenital malformation, the causes of their development remain unknown. And also we need to classify the congenital malformation of gastrointestinal tract by its localization. On this picture, present their main organs of gastrointestinal tract, and in each organs may happen their congenital malformation. So there are some characteristics of most of them. For example, congenital pyloric stenosis, prevalence 1 case for 150 male newborns, or 1 case in 70 or 150 females newborns. The type of inheritance, additive polygenic forms, and you may to see hypertrophic pyloric stenosis present on this photo, and find clock in patient in newborns with their congenital pyloric stenosis. The next examples are Hirschsprung disease. A multigenic neonatal intestinal obstructive syndrome that is correctable by surgery. It is genetically heterogeneous and clinically variable, ranging from several with a high neonatal lethality to moderate in late infancy and mild in childhood. Frequency of occurrence, 1 case in 5,000 newborns. The sex ratio is approximately 4 males to 1 females. In case with the Hirschsprung disease, we need to detect the mutation in several genes. The main candidate genes, RET proto-oncogene, endothelin receptor gene, glial neurotrophic factor gene, endothelin 3 gene, neurorenin gene, endothelin converting enzyme 1 gene, and etc. So on this slide, you may just see the main genes which are associated with Hirschsprung disease, alleles or variants. You may just see that the main gene with the pivotal role is red, for example. On this slide, on this table, you may just see the short name of the gene, OMIM number, its localization in chromosomes, and some detailed characteristics. You may make a photo of this chart, and when you will be had the topic in practical class, you may use this information for illustration your answers. The next form of the hereditary diseases of gastrointestinal tract, typical common form, peptic ulcer disease. Peptic ulcer disease. It occurs when open sores or ulcers from the stomach or first part of the small intestine. Chronic polyetiological recurrent disease, the development of which is based on complex nerves, hypothalamic pituitary, adrenal, and local gastroduodenal mechanism, leading to changes in trophic processes in the mucous membrane of the stomach and duodenum. Epidemiology. Ulcers affected about 4 million people each year. More than 40,000 people have surgery because of persistent symptoms or problems from ulcers. About 6,000 people due to ulcer-related complications. Prevalence of peptic ulcer depending on their age and sex. Ulcer can develop at any age, but they are rare among teenagers and even more uncommon in children. Duodenal ulcer occur for the first time usually between the ages of 30 and 50. Duodenal ulcer occur more frequently in men than women. Stomach ulcers are more likely to develop in people over age 60, and stomach ulcers development more often in women than men. General classification of peptic ulcer disease include first, multifactorial form of peptic ulcer develops as a result of interaction between multiple genetic and environmental factors. The additive polygenic model of inheritance is characteristics for this form. The multifactorial form is mostly common in various populations. Second form, monogenic peptic ulcer, develops as a result of single gene mutation that is responsible for gastroduodenal ulceration. These forms are inherited as their autosomal dominant characters. There are two main monogenic forms of peptic ulcer. Normopeptinogenemical duodenal ulcer and hyperpeptinogenemical duodenal ulcer. The next, third form, peptic ulcer due to hereditary syndromes. There are gastrocutaneous dysplasia, Goffin-Seros syndrome, essential tremor, nystagmus duodenal ulceration, and other. Moreover, the gastrointestinal ulceration can arise as a result of many common diseases, such as cystic fibrosis, hyperparathyroidism, carcinoid syndrome, hereditary alpha-1 antitrypsin deficiency, etc. Fourth form include immunological peptic ulcers. Thus, peptic ulcer is genetically heterogeneous pathology. Identification of independent genetic forms of peptic ulcer is necessary for developing the approaches to the differential management of disease, prevention, and forecasting of complications. The next classification depends from the genetic or pathogenesis. The first form is elated gastric or duodenal ulcer disease in adults. Second form, combined gastric and duodenal ulcer disease. The next, juvenile children form of duodenal ulcer disease. Peptic ulcer due to inheritable diseases, normopepsinogenic duodenal ulcer, hyperpepsinogenic duodenal ulcer, and immunological forms of duodenal ulcer disease, and peptic ulcer associated with other chronic diseases like secondary ulcers. There are a lot of environmental factors may cause this disease. First of all, lifestyle. These factors include cigarette smoking, food and beverage containing caffeine, alcohol, and of course, sexual stress. So, environmental factors such as acid and pepsin also includes non-steroidal anti-inflammatory drugs also may cause their disease. We know there are a lot of scientific studies about the Helicobacter pylori infection and genetic factors. Includes family history of peptic ulcer disease and gastritis, courage of the first or zero blood group and Rh negative blood groups antigens, insufficiency to secretes of ABH antigens in stomach, deficiency of protective phogoglycoproteins in gastric slime, disability to field test of phenylthiocarbamate, and courage of B5, B22, usually antigens, and hyperpepsinogenemia, also such as genetic factors of peptic ulcer, and hypergastrenemia. So, familial aggregation of peptic ulcer. Early studies recognize a familial aggregation of peptic ulcers displaying complex patterns which were interpreted as polygenic inheritance. Approximately 22 or 50% of duodenal ulcer patients have a positive family history for duodenal ulcer, compared to 5 to 15% of non-nuclear subjects. Concordance of peptic ulcer is more common in monozygotic than dizygotic twins, suggesting a genetic components to the familial aggregation of risk. However, incomplete concordance of ulcer in monozygotic twins indicates that monogenic factors are also operative. So, we need to learn the special genetic markers. We talked with you about the first blood group, Rh negative factors, and etc. On this slide, you may just see the main genes are associated with accessibility to peptic ulcer. there are, for example, pepsinogen, C gene, endothelial nucleoxide synthase gene or NOS3 gene, interleukin 1RN gene, and epidermal growth factor gene, and etc. Also, genes are associated with the susceptibility to peptic ulcer, includes their genes of involves into metabolism of the xenobiotics. It is their genes of the cytochrome enzymes, cytochrome P450 genes, and microsomal epoxide hydrolase gene, glycogen as transferase P1 gene, much drug resistance protein gene, and mutation in this gene associated and involves into the pathogenesis of the peptic ulcer. So now I think that we need to have the break 10 minutes, and after that we will be continue. Do you hear me, guys?
Yes,
Do you
but
hear me?
Yes, ma'am.
Yeah?
Yes,
Okay,
ma'am.
10 minutes, and after that we continue. So please, let's continue. Do you hear me, guys?
Yes, ma'am.
Yes, madam.
Okay, so the monogenic form of peptic ulcer are characterized by the following main features. Monogenic form of peptic ulcer manifests generally in onset in 25-30 years than polygenic forms. Peptic ulcer appear in the pyloroduodenal zone due to increased secretion of hydrofluoric acid in the stomach. The next, acute onset of disease, quick progressing disease, often complication, no positive effect from traditional management. Also, identical prevalence of disease among men and women. No family history for gastric or duodenal ulcer disease, chronic gastritis, or other common gastrointestinal diseases. And high genetic risk of peptic ulcer for first-degree relatives or purposes. So it is the main characteristic for the common monogenic form for all multifactorial disease. Also, we told with you on previous lecture, pathology, and for example, today about the monogenic form of peptic ulcer. For example, of monogenic form of peptic ulcer disease, we need to explain about the multiple endocrine neoplasia type 1 and Zollinger-Ellison syndrome. So, multiple endocrine neoplasia type 1 is an autosomal dominant disorder characterized by varying combinations of tumors of parathyroids, pancreatic islets, duodenal endocrine cells, and the anterior pituitary with a 94% penetrates by age 50. Less commonly associated tumors includes ergot carcinomas, lipomas, angiofibromas, thyroid adenomas, spinal cord ependymomas. So, MAN1 gene mutation on the 11th chromosomes. Also, there are some genes which are associated with another form and other forms of multiple endocrine neoplasia includes MAN2A form and MAN2B forms. The number, you need to see presence here, you need to read more detailed characteristics of these monogenic forms, and both of which are caused by mutation in the red gene, and MAN4 also type, which is caused by mutation in the CDKN1B gene. Another examples, multiple endocrine neoplasia, Zollinger syndrome, systemic mastocytosis is a disease, presumably with autosomal dominant inheritance, ectodermal gastric dysplasia with autosomal dominant inheritance. So the next question of our today's and congenital diseases of the respiratory system. From the genetic point of view, diseases of the bronchopulmonary system can be divided into the following forms. Monogenic pulmonary diseases. Monogenic forms includes Muir-Koch syndrome or tracheobronchomegaly, Williams-Campbell syndrome, primary ciliary dyskinesia, primary pulmonary hypertension, idiopathic pulmonary fibrosis, bronchiectasis, Goodspatcher syndrome, idiopathic pulmonary hemosiderosis, and etc. The second forms, inheritable diseases affecting the respiratory system. Typical cystic fibrosis, inheritable immunodeficiency disorders, Marfan syndrome, Ehlers-Danlos syndrome, Klinefelter syndrome, hemophilia, and etc. For example, in case with the detection of Marfan syndrome, may developments and etc. The next, third form, multifactorial diseases of respiratory system include primary pulmonary emphysema, chronic obstructive pulmonary disease, bronchial asthma, chronic bronchitis, exogenic allergic alveolitis, and other ones. The fourth form includes lung malformation. Most forms are akinesia, aplasia, hypoplasia of lungs, congenital lobar emphysema, congenital cystic adenomatous malformation, pulmonary sequestration, bronchogenic cyst. So, more detailed characteristics of the congenital lung malformations are a family or structural development abnormalities of the lungs that arise from an error in the embryonic development of the lungs. The lung normally develops from one kind of tissues that becomes the airways arising originally from the upper digestive system, and another kind of tissue that becomes the blood vessels and connective tissues of the lung. These two kinds of tissues must communicate clearly with each other to form a normal lung. Errors in communications can lead to one or more of these malformations. On this picture, you're going to see the degree of lung malformation, lung akinesia, absence of lung aplasia. You're going to see the rudiment of main bronchus presence, their secondary bronchitis and alveolus are absent. Lung hypoplasia, underdevelopment of small bronchi presence here, development of small bronchi. Just radiographs, for example, may show for us upper lobe akinesia and the right lung, or total akinesia of the right lung. And on this picture presents the rudiment lobular bronchus. Polycystosis of lungs, a number of small cysts in lungs present in these photos. Another examples of the congenital lobar emphysema. This malformation is characterized because of poor development of cartilage in the large airways. The airways are being very soft. This causes them to collapse and trap ever increasing amounts of air in the lungs, which air inspiration. They usually occur in the upper lobes on either side of the chest. Emphysematosis areas in upper lobes of right and left lungs present on these photos. How is the diagnosis of lungs malformation made? Prenatal or before birth, very important early diagnosis, of course. Many lung malformations are diagnosed on routine prenatal high-resolution ultrasound. Lung malformation can be seen in earliest 15-20 weeks gestation and are usually incidental indents. On the rare occasions, particularly if the malformation is acute large, they can cause the fetus to look very swollen, like fetal drops. They are worsening feeding. Almost always the only action needed if a lung malformation is identified is prenatal consultation with a pediatric or surgeon comfortable and familiar with these types of diagnosis. These lung malformations are usually isolated findings, not genetic in a region and not part of a syndrome. Postnatal diagnosis after birth. Only those malformations large enough to cause the symptoms will be diagnosed after birth because a well-appearing child would have no reasons to be evaluated any further. Newborns infants who are symptomatic form a large malformation with demonstrative hydrops, heart failure, or respiratory failure. Older children can present with an infection in malformation that looks like a lung abscess, bronchogenetic cyst, or repeated pneumonia in the same locations. So, in either case, the diagnostic evaluation is derived from a combination of chest X-rays, computerized axial tomography scan, or magnetic resonance imaging. Pulmonary function testing and echocardiography may complement the evaluation. Now let's give more detailed characteristics of the slide. You might just see the pulmonary manifestation of the uh, pulmonary form of cystic fibrosis, multiply infiltrates due to obstruction and bronchiectasis occurring in the small bronchi. Let's give characteristics of the thumb monogenic pulmonary disease. First of all, primary ciliary dyskinesia caused by compound heterozygous mutation in dynein axonal intermediate chain 1 gene protein, which localizes on chromosome 9. Primary ciliary dyskinesia is a genetically heterogeneous autosomal recessive disorder resulting from loss of function of different parts of the primary ciliary apparatus, most often dynein arms. Cartagonell syndrome is characterized by the combination of primary ciliary dyskinesia and situs inversus and occurs in the approximately half of patients with ciliary dyskinesia. Cartagonell syndrome is one of the clinical conditions caused by cilia not working properly. This syndrome is caused by disordered ciliary movement that results in infective mucus transport in the lungs. The disease is called primary ciliary dyskinesia because of the disordered ciliary movement that is a common thread that unites Cartagonell syndrome main clinical manifestations. Main clinical picture. Cartagonell syndrome referred to the clinical triad of situs inversus, bronchitis, and male infertility. Common presentation includes neonatal respiratory distress, recurrent episodes of otitis media that may lead to deafness, situs inversus, bronchiectasis, digital clubbing, nasal polyps, and male infertility. Diagnosis of Cartagonell syndrome includes symptoms may suggest cystic fibrosis, but the sweet test is negative in this case, with Cartagonell syndrome. The patient may present with infertility but have a normal sperm count. The diagnosis is suggested by the clinical presentation of the patient and confirmed by electron microscope examination of ciliary ultrastructure in the nasal epithelial cells. On this slide, on this photo, you may to see the dextrocardia and thickening of a middle lobe of left light having 3 lobes. And heart is turned to the right side, bronchostasis and atelectasis of middle lobe of left lung present on this photo. So next monogenic form, idiopathic diffuse pulmonary fibrosis. Damage to the pulmonary interstitium with the subsequent development of pneumosclerosis and respiratory failure, hypertension of pulmonary circulation, and pulmonary heart. It has autosomal dominant type of inheritance. The main gene which associated with this idiopathic diffuse pulmonary fibrosis named surfactant lung associated protein I2 or SFTP2 gene and mucin5 subtype B tracheobronchial gene, localization on 11th chromosomes. Pathogenesis of this disease, originally idiopathic pulmonary fibrosis was viewed as a modeling inflammatory response that ultimately led to chronic lung injury with subsequent fibrosis. Never insights suggest the idiopathic pulmonary fibrosis results from the sequential acute lung injury. The resultant Wood-Helling response to this injury culminates in the pulmonary fibrosis. Several interaction factors that modify the fibrotic response include the genetic background of the patient, the predominant inflammatory phenotype, and the environmental inflammatory triggers such as cigarette smoking, viral infection, and respirable toxins. The next monogenic form, primary pulmonary hypertension. Primary pulmonary hypertension, a rare, often fatal, progressive pulmonary vascular disease characterized by increased pulmonary vascular resistance and persistent increase in mean pulmonary artery pressure, resulting in right ventricular hypertrophy and right ventricular failure. Pathological signs include narrowing and thickening of small pulmonary vessels and plexiform lesions. The prevalence is approximately 1 in 100,000 to 1 in 1 million people. The type of inheritance is autosomal dominant with low penetrance, affecting more women than men. The phenomenon of anticipation is characteristics. What does mean anticipation? An increase in the severity of the course of the disease in the number of generations. The aggravation of the course of a hereditary disease from one generation to another. So these photos, you will see dilatation of proximal portion of dilatation of lung roots and weakening of pulmonary picture at peripheral parts of lungs. The next form, monogenic form, alpha-1 antitrypsin alpha-trypsin deficiency, hereditary disease in which lesions of the lungs, liver, and blood vessels develop. The cause of the disease is a defect in SERPINA1 gene, which encodes the alpha-1 antitrypsin enzyme.
A common complication of alpha-1 antitrypsin deficiency is primary pulmonary emphysema, hereditary disease in which lesions of the lungs, liver, and blood vessels development. The cause of the disease is a defect or mutation in SERPINA1 gene, which encodes the alpha-1 antitrypsin enzyme. Emphysema, so primary pulmonary emphysema, the next monogenic form, is a disease made by destruction in the alveoli, grape-like clusters of air sacs at the end of the smallest airways, the bronchioles in the lungs. The K-process leading to emphysema is destruction of a protein in the lungs called elastin, which is essential for the springy quality of many tissues in the body. This protein is especially important in the lungs for maintaining flexibility of the airways. So you need to see in the emphysema alteration of lungs at the initial stage of the disease. Emphysema areas in lungs, you need to see presence here, and thickening and dilatation of bronchial walls. In pulmonary form of cystic fibrosis, multiple infiltrates due to obstruction and bronchiectasis occurring in small bronchi. So the next question of our lecture, bronchial asthma. Common chronic respiratory disorder featuring episodic shortness of breath, often at night and usually accompanied by non-productive cough. Asthma is a chronic inflammatory disorder of the airways, chest, tonsils, and coughing, particularly at night or in early morning. The numerous genome-wide linkage, candidate gene, and GWAS are performed in asthma and asthma-related phenotypes have resulted in an increasing large list of genes implicated in asthma susceptibility and pathogenesis. Asthma, as an immune-mediated disease, involves a response of the body to environments, including pollutants, allergens, virus, and other pathogens and irritants. These environmental factors interact with the genetic variation to influence the development of severity of disease. Researchers are finding that specific genetic variants affect susceptibility to and the severity of asthma in different ways depending on the environments of the individuals carrying those variants and phenomena known as a gene-environment interaction. Twin studies have shown that the genetic element of asthma susceptibility with heritability of the condition is made at between 36% and 77%. Seems that more than 600 candidate genes described in more than 1,000 publications have been found in connection to asthma or an associated phenotype like elevated immunoglobulin E levels or eosinophilia, and etc. You may to see on this slide the name of the gene which involves in immune function, barrier function, and tissues response, its chromosomal localization, and its function. There are a lot of the genes are repeated again, 600 candidate genes described in the literature which associated with the polygenic forms of the bronchial asthma. And you may to see there are genes implicated in asthma susceptibility and pathogenesis, the genes which involves into the epithelial barrier, environment sensing and induction, Th2 mediated cells response and genes which detect the enzymes, tissues response. Th2 mediated cells response. Th cells mediated adaptive immune response have been widely recognized as a crucial component of allergic disease. Genes involved in Th2 cells differentiation and function have been extensively studied in asthma candidate gene-associated studies. And as one might expect, the single nucleotide polymorphisms in many of these genes have been associated with asthma and other allergic phenotypes. Genes important for the Th1 versus Th2 T cells polarization, like GATA3, TBX21, interleukin 4, and etc. have been implicated with asthma and allergy. Environmental sensing and immune detection. A second class of associated genes is involved in detection of pathogens and allergens. The genes include patterns, recognitions, receptors, and extracellular receptors, like receptor. Additional studies have strongly associated variants in the Escherichia class 2 genes with asthma and allergen-specific immunoglobulin E responses. These molecules are important in the immune response and shaping of the T cells repertoire. Tissues response. A variety of genes involved in mediating the response to allergic inflammation and oxidant stress of the tissue levels appear to be important contribution to asthma susceptibility. Some examples include the disintegrin and metalloprotease expressed in lungs, fibroblasts, and smooth muscle cells, the alpha-1 gene of type 21 collagen, leukotriene C4 synthase, glutathione as transferase, nitric oxide synthase, 1 or NOS1 gene, and etc. On this slide, you may see the most reported genes which associated with childhood onset asthma. It's localization on chromosomes, the name or top SNP, and analyze or phenotype in which we may to detect some mutation in these genes. In conclusions, today's discoveries in inflammatory only explain a fraction of the heritability of asthma. Referred to this missing heritability, thus difficult to fill void in our understanding, has become a common theme in the analysis of complex genetic traits. ESMO was genome-wide association study of increasingly greater size are performed. It is likely that additional loci will be identified that will close a portion of this gap. However, genome-wide association studies are not well suited to detection of rare variants with potentially large effects. More importantly, the development of techniques to describe gene-gene and gene-environmental interaction, which likely play a key role in the susceptibility to complex diseases, is only in its infancy. Very little is known how epigenetic phenomena, heritable but reversible changes to DNA, such as methylation, that are not detected by standard genotyping, may contribute to asthma and allergy pathogenesis. Advancement in sequencing technologies is likely to enable strategies capable of thoroughly examining to entire genome, including all genetic and epigenetic variation and their complex interaction. Eventually, we will be able to complete the journal from the genetic discovery to improvements in asthma patient care. So, thank you for your attention. If someone have questions, let's discuss it. Don't forget that today we have the last lecture in genetics medical ecology. With some students, we told in classes in genetics, we discuss these questions. With other students in future, we will be discuss this all question in genetics. And please don't forget that in the next semester, you will be have the second part of this discipline, medical ecology. And in medical ecology, we will be give more detailed characteristics of the environmental risk factors of complex pathology, and we will be give detailed characteristics of the influence of individual climatic factors into the human health. So, do you have a question about today's lecture?
No, ma'am.
If no, good luck to you. God bless.
Thank you.
And see you on the